So for our second speaker for today and tomorrow, we're excited to have Alison Hill. Alison is an assistant professor of biomedical engineering at Johns Hopkins University and a core faculty member at the Institute for Computational Medicine and in the Infectious Disease Dynamics group. She received her bachelor's of science in physics from Queen's University in Kingston, Ontario, and her PhD in biophysics and medical physics through the Harvard MIT Division of Health Sciences and Technology, and a master's in public health from the Harvard School of Public Health. She's a previous recipient of the NIH Directors for Independence Award and the World Economic Forum's Young Scientist Award. Her research team develops mathematical models and computational tools to help understand, predict, and treat infectious diseases, with particular focus on HIV and AIDS, which we'll talk about today, SARS-CoV-2, COVID-19, and drug-resistant infections. And on a personal note, she's an amazing postdoc supervisor, so I'm very excited to have Alison here to talk about her work and all that.
Great. Thanks, Morgan. And thanks so much to you and the rest of CRM for the invitation to speak here today. So, yeah, I'm going to give a bit of an overview today on how we use mathematical models to better understand and make predictions about treatment strategies for HIV infection. And I'm totally fine if anyone has questions at any time throughout the talk, like feel free to interrupt me. So overall, like big picture, in our lab, we focus on developing models, mechanistic mathematical models to understand biological systems. And we focus in particular in infectious disease dynamics. And we think about the dynamics of infections both within individual patients and across populations, and how processes at one scale affect those at the other scale, which is very important for disease evolution. And so in general, I think particularly if you were at Ruth's talk, a lot of the way that we think about models is similar. So often we use models as a tool, kind of integrate previous biological information of different sorts to come up with either hypotheses about why we see certain observations in biology, or to make predictions about something we might see in a scenario we haven't yet conducted, or kind of make recommendations personalized for individuals or particular populations about intervention scenarios. And ideally, we have a feedback between what a prediction of a model is and what kind of biological or clinical or epidemiological studies can be done to help improve our understanding and then encode that in future models. So since I'm going to be talking about HIV today, I wanted to first give a little bit, sorry, I'm having some weird side issues, a little bit of an overview of HIV for those who might be a bit newer to this disease. So, we've all lived through one pandemic, but for many of us, COVID was not the only pandemic of our lifetime. And before that, I think probably the biggest major one was HIV, and it's still continuing to this day, and is still a pandemic as much as it ever was. So this virus and the disease, AIDS, that it causes was first identified in the early 80s based on like a syndrome of individuals suffering from what seemed like sort of severe immune deficiency syndromes and having infections and cancers related to that, which eventually led to the identification of this new virus, HIV. Pretty rapidly after that, the virus kind of spread around the entire world, and to this day is still increasing in prevalence every year. We know now that actually HIV was in human populations probably since the turn of the 20th century, and it actually arose from a zoonotic transmission event from a virus that is like naturally occurring in primate populations. And what we know as HIV is actually many different viruses. We know at least 3 separate monkey species that transmitted HIV into the population, and some of them have transmitted different strains multiple times. So really multiple zoonotic origins of this virus that we now call collectively HIV. And today, around 40 million people around the world are living with HIV, around 1 million deaths a year from HIV. For many years, decreased a little bit in the last few years. Still new infections occurring every year, kind of outpacing that, so the infection's growing. And we will hear some more optimistic notes soon about what is turning around this epidemic. So within individuals, what is happening in this disease? So when someone acquires HIV, usually through sexual transmission, can also be from perinatal events like mother-to-child transmission, or from contaminated blood products, the infection really rapidly progresses through what we call an acute infection stage, where you get really high levels of virus in the body, and you might just have typical viral infection symptoms, kind of like flu-like symptoms, fever, fatigue, things like that. And then after that, the infection is sort of partially controlled, but reaches something that we call a set point. So some level of virus still quite high in the body. It's maintained be for many years, even decades. But eventually, without treatment, pretty much 100% of people progress to the disease state, which is called AIDS, which is 100% fatal eventually without treatment. So kind of going down to the cellular level, this is an infection mainly of the bloodstream, also the lymph system, and within our blood, a particular type of white blood cell called a CD4 or a helper T cell is the cell that the HIV virus primarily infects. As a result, one of the main sort of consequences of HIV infection, other than these really high levels of virus in the blood that we see, is a depletion of our CD4 count, or the level of these particular T cells, what type of white blood cell in the body. And that consequence is actually what causes eventually the disease, AIDS, and all these other types of infections that can come in and take over when the immune system is just not able to fight them off. Within the cells, when the virus infects them, it has some unique parts to its life cycle. And those are actually really important for all of these issues with controlling the epidemic globally and with the new type of treatments that we might use here. So I'll just point. Okay. So when the virus enters cells, it actually carries its genome in RNA form, and it has to reverse transcribe, meaning reverse that RNA and turn it into DNA when it enters the cell. And when it does that, it actually integrates its little piece of DNA into the human DNA in our chromosomes in the nucleus of human white blood cells. And then more copies of that virus genome can be produced, turned back into RNA, can make viral proteins, and we can assemble sort of thousands of these viral particles in any given cell that's infected. And kind of the useful things to know about this process of going backwards from RNA to DNA is quite error prone. So HIV has a pretty high mutation rate compared to a lot of other viruses, though most viruses have pretty high mutation rates. This is extra high. And also the fact that it integrates its genome into our DNA is a really important thing that makes the disease difficult to control, which I'll get to in a second. So luckily, we now actually have many different drugs that specifically target HIV infection and can actually prevent this previously guaranteed progression to AIDS and then the fatality that came along with that. So there are many different classes of HIV drugs, and basically what that means is they just block different targets of the viral life cycle. So kind of early drugs, the most commonly used drugs still today, are actually preventing this reverse transcription process. But we now have drugs that block this integration process. They block these viral particles from kind of assembling correctly. They might directly block entry into the cell. But overall, their main effect is the same, that they lead to a dramatic reduction in the viral loads within individuals who are taking them. And as a result of that, they can both dramatically increase the life expectancy of people living with HIV and also reduce dramatically the chance that that individual would transmit HIV to someone else, kind of by any means. So this has been a huge change in the epidemiology of HIV. So now in the last kind of 20, but even mainly the last 10 years, the number of people on receiving some type of antiretroviral therapy, ART is like a short name for that therapy, has been increasing every single year, to the point that every time I have to update a talk I give on HIV, I get excited because the numbers, the curves are kind of getting closer. So now almost 30 million people around the world are on some type of antiretroviral therapy, which is like an unimaginable achievement for science and public health. And I'll tell you like why that is such an achievement, because there are many challenges here. And the timeline here is a bit interesting, because actually antiretroviral therapy was discovered to some degree in the 80s, some of the early drugs. It wasn't until kind of early to mid 90s that we had really effective drugs, kind of the best ones that we're mainly using today, and that we had enough of them that we could really use them in combination, which was a really important thing to get them to work long-term for HIV. And we'll talk about why that is. But that still wasn't enough, because that was around this time period. As you can see, we still had a long time when very few people were benefiting from this. And it's really a huge logistical challenge to provide therapy to this many people all around the world, many of whom are living in sub-Saharan Africa or in other parts of the world where healthcare infrastructure is very limited. And actually, a lot of this progress is really due to a lot of advocacy, a lot of public health work, and a lot of donors, both from private agencies like the Gates Foundation, which funded a huge amount of this, as well as governments all around the world. Surprisingly, PEPFAR, which was an initiative by George Bush in the US, was one of the main catalysts of this original increase. And actually, a lot of that funding is in threat right now. But this has really eventually led to huge improvement for people around the world living with HIV. So some simple but very useful math can help us actually understand some of the important aspects of HIV disease without therapy and the impact of therapy. So models were actually used pretty early on in the understanding of HIV, and those models form still to this day the framework of a lot of the work that is done. So what I'm showing here is really we call the basic viral dynamics model for HIV. So this is sort of like the simplest ingredients going into a model for what happens in an individual who has HIV. And so the main thing that this model tracks is the target cells of the virus. So these are mainly those CD4 T cells. They're natural population dynamics. They're being produced, and they have some sort of finite lifespan, so they're dying, just kind of even in the absence of infection. And then in contact with virus, those cells can become infected. When they do, they have a much higher death rate, and then they, after some time, can start releasing more virions into the bloodstream, which can then infect further target cells. So this model can be represented with a pretty simple system of differential equations. Importantly, it is a nonlinear system because this infection process requires contact between both virus and target cells. And not too hard to kind of look at the outcomes of this model. And even though it's so simple, it really recapitulates some of the main facets of HIV infection. So you can see here, you have this really rapid exponential increase early in infection, this huge peak that occurs, and then some eventual stabilization to this more steady state, or as we call it, set point, accompanied by this large decrease in CD4 cells. So this model, even in its simplicity, like one important thing that it showed, for example, is that this decrease of this peak doesn't need any complicated immune response to be explained. There's no immune response in this model. Actually, it just turns out that the difference in time scale between the turnover of these target cells naturally, which is quite slow, and their death rate when they are infected, which is much higher, can lead to this sort of overshooting of the peak. It has since been shown that actually to get as big of a drop as you see in natural infection, you do need some immunity, but the basics are there. But then more importantly, actually, when early studies of HIV treatment started, we learned something else from this model very useful for studying infection. So this is data from relatively early study of what turned out to be one of the first pretty effective anti-HIV therapies. And this is showing the change in viral load of a set of patients who received this therapy. And you can see everyone's viral load drops very dramatically over the course of just a few weeks. And this was the first time this type of dramatic change had been seen. And in this model, in the simplest way that you could model therapy is saying that this blocks the infection of new cells. And when you do that, in this sort of model, you see this decrease. Basically, you're turning this nonlinear system into a linear system, and you can actually work out mathematically what that decrease should look like. It can theoretically depend both on the clearance of free virus particles themselves and on the natural lifespan of these infected cells. But basically, this decay rate will be the slower of those two processes. Virus itself is just protein floating in the blood, tends to be cleared very quickly. So really, this decline should reflect the lifespan, so the death rate of those infected cells. So what that said, because this decline was so quick, it corresponded to a death rate of around a day. So that's suggesting that infected cells are only living around a day. So why is that, so interesting? Well, we know that the whole time course of HIV infection is very long, right? It takes people many years or decades to actually progress to this deadly stage of the disease. And for a long time, people thought maybe that meant the infection itself is very slow. There are other viruses in the body that we have for life that have a very, very slow turnover. But actually, if infected cells are average living like a day, that means every new day you must be replacing all the infected cells in the body that are generating all the virus in your body. And if you work out just based on like the concentration of virus in someone's blood, like how many infected cells that is, it's like billions. So this is an extremely rapidly progressing infection, despite the fact that there's a separate time scale to when disease develops. So this was important to help us understand like what is actually going on with this infection. It was also important because if there's that much turnover in viral population, it means there's so many opportunities for the virus to produce mutations that could compromise the ability of therapy to control it, which is something that we'll see in a second. And so you might have noticed that this model has some familiarities with things like an SIR model that we've now heard lots about used to model disease epidemics at a population level. And actually, they are quite similar. This idea that you have sort of some resource, which is the population you are trying to infect, and then contact between an infection and that sort of host or resource. So it is quite similar. And similarly, these viral dynamics models have these type of threshold parameters really analogous to a basic reproduction number. Yeah.
Sorry, I have a question.
Yeah.
So in your model, there's no equation for CD4 T cells, but how?
Oh, sorry. This weirdly, oops. Okay, look at the variables. That equation's using some notation that clearly didn't get updated. These are the CD4 cells, the target cells here. Just a mistake on the next slide in terms of the notation, but that is the CD4 count. So yeah, weird. But basically, this reproduction number depends on all the parameters in the model, and what it corresponds to is this average number of secondary infections in terms of infected cells produced by, on average, one single infected cell when all the other cells are uninfected. So it has analogous meaning, and similarly, it is what we call a transcritical bifurcation in the system. So when it's greater than one, the infection can grow. When it's less than one, the infection will eventually will decrease and eventually go extinct. And if it changes between which of those two steady states is the stable one. And so when R naught less than one, that's basically what therapy should be accomplishing. And what we saw even in that early trial, that leads to infection levels decreasing. So that sounds great. You would imagine, okay, we just give people therapy for long enough and then the infection's going to be cured, just like, you take antibiotics, eventually you stop taking antibiotics and then that's the end of it. It turns out that that is not what happened with HIV. And what we see is pretty much everyone, whenever we stop antiretroviral therapy, no matter how long it's been, a week, a year, 10 years, infection always comes back. It comes back quickly and it comes back to high levels, close to where it was before treatment. So this is called viral rebound, and it's something that makes HIV treatment very complicated. And so what is happening here? Like where is the infection coming from? So it turns out that there is a population of cells that still have the virus integrated into their genome, but are not turning over at this fast rate that those previous studies were suggesting. And that's actually because of the type of cell HIV infects. This is a type of immune cell that can go into kind of dormant, hibernating, quiescent, resting. We have lots of word for this, but it's a state that actually preserves the memory of your immune system. That's what it's supposed to do. You know, these cells normally, their job is to fight off infections like HIV or any other infection. So every once in a while, they'll go into this dormant state to stick around for a long time. So you remember infections that you had when you were a child, for example. And if they happen to do that when they're infected with HIV, now there's a pool of HIV infected cells that can live for decades. And it's those cells persisting. We think most people receiving HIV treatment probably have something like 1 million of these cells in their body, and they can at any time wake up. It's just spontaneous. Some of them are waking up every day, release virus, and restart this infection if therapy is starting. So this is a huge barrier to HIV treatment. It means we can't cure infection. People who take therapy have to take it every day for their whole life. And whenever they stop it, they're at risk of this rebound happening within kind of maximum a few weeks. So math can actually help us understand what's going on here and what we might do about it too. So for a while, a big push in the HIV field was to try to get rid of these latently infected cells, or to at least like reduce this population. I said it's 1 million. What if we can get it down, to 100,000, 10,000, thousands by some means? Can we target these cells somehow and reduce this pool? So it's just, maybe we can just make it so unlikely that this would happen. And there are a lot of different means people tried for doing that, whether it's pharmacological or immune or by like gene therapies, all kinds of things. So the question is that a feasible solution to try to cure HIV? And by cure HIV here, we just mean that like practically, most people wouldn't see a rebound when they stop therapy. And to add to this, that doesn't necessarily mean you would have to get rid of every virus. We probably have lots of viruses floating around our bodies. Normally, our immune system controls them. The immune system is really not good at controlling HIV, but sometimes just by chance, it's just unlikely to happen. So this is something that we studied a while ago to try to understand. So the idea here is you have this sort of pool of these latent cells, someone's on combination therapy, some other type of intervention dramatically reduces this cell population by some amount. And then we stop therapy, and what happens? So if we think about every single cell that's in this latent reservoir population and sort of a few different things that can happen to that individual cell, it might just die. Those cells do not have an infinite lifespan, or it might reactivate, start producing virus, lead to some cluster of infected cells if that cell itself doesn't die. And that could proceed kind of all the way up to eventual viral rebound. If enough infection events happen, we get the degree of virus that can actually be detected. Or if kind of none of these cells manage to complete each of these steps without kind of dying off, then that would be what we would call a cure. Or if they don't do that in the lifespan of a typical individual. So this is a type of, not too complicated, but sort of multi-type branching process model that we can use to understand this. And there are lots of different types of data we can use to try to actually work out the different parameters here. And when we do this, we can get these type of sort of survival curves, which what they show here is depending on the degree to which you reduce this pool of latent virus compared to what a typical individual would have, how long or how many people are still sort of suppressed is based on the amount of time that we've had them off therapy. So what this means here is that, let's say we reduced by a factor of 10 or a factor of 100 the size of this pool, what the survival curve suggests is that most individuals still within 100 days would rebound. So that's a few months. So that is not really a helpful cure. Even a 99% effective drug here would really just delay the inevitable by a few months. When we get up to these regimes where we would reduce the size of this latent pool by 1,000, 10,000, we start to see the fact that some people could go for years without rebounding, but many of them would still rebound, and it would be totally unpredictable when this would happen. This is really just like a waiting game for the random process to happen. So this suggests that this is really a very difficult way to try to cure HIV. When we first suggested this, I think people thought this was like a little bit crazy and unlikely, like, oh, virus is just going to wait around and then 3 years later, you're going to see someone rebound. But actually, this ended up happening a bunch of times. So this is a bit of a weird plot. This shaded area here is basically showing things that with the model would suggest are possible for the relationship between how long someone is off their therapy until they rebound and how much you've reduced the reservoir size. And basically all these red dots or points here are actual people. And what it means are these are different times in which individuals who receive some type of intervention that dramatically reduced their latent pool, at what time they actually ended up having a viral rebound. And the reason these are not dots, but like points, is because we don't actually know how big their latent reservoir is. It's very hard to measure. Usually we can just tell it's below some limit of detection, which is like the lower values here. So there are many individuals, some who received stem cell transplants, some who were treated so early there wasn't even time for latency to form, and some sort of more complicated situations. But many times people went many years. Everyone thought they were cured of HIV or wished they were, and then the infection came back, and it came back very rapidly, and it came back in a way that genetically suggests a single cell reactivating with virus caused that reactivation, as opposed to like all the diversity genetically we normally see in HIV infection. So this is another major barrier to a potential way of curing or controlling HIV infection. Yeah.
Arrow horizontal.
Yeah. So like, this is the one person, well, at this time, there's more now, who might have been cured of HIV. And so this arrow actually means it was longer than 10 years and they didn't rebound. So this individual actually passed away eventually of other causes, but went 10 years without being rebound. And now there is someone else, not so long, but like a few years, who still hasn't rebounded. But these people, there's kind of something else going on here. They actually received stem cell transplants. That's not something we're like considering to do to every person with HIV, extremely risky and expensive, but they had a type of cancer that required that as the treatment. And when they found a donor to give them their stem cell transplant, they found someone who carried like a human genetic mutation that made that human's cells quite resistant to HIV. And they gave that as a part of the stem cell transplant. They also had to like have a HLA match and all that, super complicated. But as a result, this individual, it's probably not just that you reduce this latent pool so much and by chance nothing reactivated. It's probably also because they are now like resistant to HIV infection. And there's another case from a group in London who did a similar procedure and had a similar result. So that actually suggests that protecting those target cells is what's conferring the cure in those individuals, not like getting rid of this latent pool. So that is not the only kind of barrier to treating HIV. Another barrier, we have this antiretroviral therapy. Can we just like eventually give it to everyone and eventually like they will have HIV, but they will be under control. They can't transmit, they won't progress. That would be great, but that's very difficult to do. And one of the most difficult things with antiretroviral therapy is just being able to take those drugs every day for the rest of your life. Most of us, if you really think about it, are quite bad at taking, whether it's your vitamins, your medication, whatever else you committed to doing. And there are other like logistical challenges like supply chain or stigma that make this difficult for people living with HIV. So adherence to therapy is another big issue. And related to that, individuals, despite being on drugs, can actually have this viral rebound due to the evolution of drug resistance. We know that this is highly associated with treatment non-adherence, but in a complicated way that I'll discuss a little bit about. But basically, lapses in adherence can lead to evolution of resistant strains that can eventually be selected and cause the virus to grow despite therapy. So this evolution of drug resistance is a really big issue for treatment around the world too. It's not like to the state of, all HIV isn't drug resistant, but it's something that makes treatment fail for some individuals, and is something we would like to reduce. So just to show some sort of pictures of this drug resistance issue, this is a graph from a really early study of HIV treatment with just a single drug. It was not a great drug. These are the viral levels, and you can see that very quickly viral levels went back up despite being on this treatment. So everyone rapidly evolved resistance to these therapies. Nowadays, when individuals are on combinations of therapies, if we look at the years that they've been on therapy, and then how many people in a cohort are resistant to one or two or all of the drugs that they are on, that is continually increasing, and the significant numbers of individuals, even in, this is a study from Canada of Canadians, mainly in British Columbia, even in that sort of idealized setting, a lot of drug resistance is evolving. If we look at other more resource challenged settings, this was a study from 2017. Things are a tiny bit better now, but hasn't been a ton of work on this over the COVID period. These are individuals in different countries, and what % of the population is carrying resistance to these different drug classes. And just, I know this is hard to see, this is like 10% here, 20%, 30%, like this is a lot of people. And we are even seeing now that resistance is being transmitted between people. So people who have never received treatment themselves, but got HIV, they're already drug resistant because it's spreading between individuals. So that's what the study is showing, how many people are resistant to this major drug class who have never received treatment themselves. And so, that's at around these 10, 15% levels. So we've spent a lot of time, Yeah.
The pre-treatment resistance.
Yeah.
So is this saying that there's more treatment resistance to NRTIs in Canada?
Yeah, so I mean, I think this is complicated, as you can tell. These are like combinations of different studies in Southern Africa. This was one study in Canada, and there's a lot of uncertainty here, but there are reasons to think that, in a sense, the more treatment, the more % of people in the population that are getting treatment, the more selection there is for the virus to become drug resistant. And so the more chance, if you just randomly get infected with HIV from anyone, that might be a drug resistant strain. So it could be, that would make sense, right? The % of people who have HIV in Canada who receive like good antiretroviral therapy is higher than that % in Southern Africa. So that would be possible. I mean, yeah, of course, in a sense, more treatment may equal more resistance. That doesn't mean we shouldn't give treatment, because we also just want to control infection. So yeah, we are interested in what is driving resistance and what are ways to think about controlling this. And one of the original things that got us interested in this question was this sort of puzzle. So I told you that people not being perfectly adherent to their therapy was something that was associated with the emergence of drug resistance. So if I made a graph like this that showed this is how adherent somebody is to their therapy, like what % of doses that they were prescribed did they take, and how likely is it that they become resistant? Like, just imagine for yourself in your head for a minute, like what shape would you expect a curve that might appear on this graph to have? And the good thing is, no matter what you said, actually you are right. So there are certain classes of drugs that the more adherent people were to therapy, the more likely resistance was to emerge. That kind of makes sense by this population argument I was saying about potentially Canada versus Southern Africa. More drug, more selection, more resistance. Okay, but then there were other classes, other class of drugs where we seem to see the opposite, who were more adherent, had less chance of developing resistance. So you might be able to explain that in that, okay, to become resistant, you have to actually like make a resistance mutation. And if you're just suppressing the infection so well, there's no chance for mutations to arise. Okay, so maybe that makes sense. There are other drugs you would see kind of more of a U shape. And that might make sense too, if kind of both of these factors are somehow important. You have to generate mutations and you have to select mutations, and there's some sweet spot there. But how can we understand, like, for which drugs one of these relationships is important, and how can we kind of just make therapy better so that overall there is less chance of resistance? So the first thing that we sort of realized when thinking about this is, like, the key underlying determinant of this is how efficacious a drug is as a function of what dose of the drug you have. Reducing your adherence just reduces the drug dose that you have. And so during my PhD, I worked with 2 other graduate students to try to understand this problem. So at the time in the laboratory of a close collaborator of ours, they had developed this very cool assay to actually characterize HIV drugs in a really realistic way. They could actually take samples of blood that contained CD4 cells from just healthy volunteers. They could grow it with this virus that you could track, and then they could really measure, as a function of the drug concentration, how much was infection decreased. And they could do that for, like, the non-resistant, that's what we call wild type form of HIV. That's these blue curves. And they could do it for different resistant strains. These are 3 example ones. And there was a lot of really interesting findings here, including that, depending on the drug, the level of drug that you actually needed to effectively get virus to some amount of suppression varied quite a lot. Sometimes these curves were quite shallow, sometimes they were quite steep. And for different mutation strains that were kind of resistant strains, actually, it just shifted these curves. So the resistant strains were not, like, totally immune to the drug being there. They just moved this curve, and some of them moved it by a lot, some of them not so much. So we thought this was really a missing piece to perhaps being able to predict, if someone has some level of adherence, they get some dose of drug, depending on how much they're taking, that changes that dose, and then that might determine what the replicative ability of different strains of the virus is. In order to do that, we need to think of a way to put all these things into these sorts of modeling frameworks that I talked about. And so the first thing really is to expand this viral dynamics model that I showed you before in a way that we can track different strains. So here I'm just tracking 2 strains, a wild type and a resistant strain, wild type resistant. And there's a few things we need to take into account here. One is that now this infectivity of the virus depends on how much drug there is. The second effect is that you can get mutations. It's not really shown here, but basically when a virus infects the cell, it might mutate to make a different strain. So resistant strain, oh, sorry, this is a typo. It's supposed to be that. Resistant strain is produced based on some mutation rate. And the other main thing is just the baseline fitness of resistant strains and wild type strains, their ability to infect cells might be different. So with this sort of model, you kind of see something like this, where in the absence of treatment, the wild type strain would have an advantage and grow to high levels. This is because if we look at these curves, when drug levels are very low, actually most resistant strains have a fitness cost. Like, they are not as good at infecting as the wild type. So if any resistant mutations exist, they're at very low levels. Just they're produced by mutation, but they're selected against. But once you start therapy, resistant strains then can have a large advantage and take over. So that they're produced before treatment, maintained at low levels. When you start the treatment, you decrease the production of mutants. You can kind of see that right here. But if any are around, sorry, it's a little manic right now. Oh, there's supposed to be something else pop out more facts. It's okay, I just said that. Sorry, something weird is happening. Okay, there we go. So more importantly, they're actually just different sort of what we call selection regimes here, where if the drug level is high enough, no strains can grow. You can always give enough drug to suppress even a resistant strain, but you might kill someone in the process. Like, this is probably a toxic level of drug in many cases. There's this intermediate regime where the mutant strain is doing better than the wild type. That's when you would be selecting for resistance. There's also low drug levels where you're not suppressing even the wild type strain. There's some overlap here where here you might be a little bit of wild type growth, but the mutant is doing better. So these are sort of drug concentrations where you're selecting for resistance. So, that helps us kind of fill in these gaps. But these curves look way more complicated for a lot of real drugs. You can have a whole complicated resistance pathways. Maybe you have to get multiple mutations, and each of them has some different dose response curve here. That's okay. That's something we can model. But the other ingredient here is that each person doesn't really just have one drug concentration. After you take a dose of drug levels can fluctuate as the drug is cleared from your body. That can mean that the drug concentration itself is something that's time dependent, meaning that you might sort of move between different selection regimes as you take therapy. Don't be angry at me. Sorry, I'm missing something. There we go. What? I'm seeing something different. Okay, let's slow down. Okay, so right, basically here's a picture of that. This is the levels of drug concentration over time. This is just for an example HIV drug and sort of a population average. When you take a pill, drug level's high. When you, between doses, it's going down. If you miss a pill, it could go even more down. And there's sort of maybe some regime here where mutants are selected. And for whole panels of mutations, you could work out kind of what are these drug regimes where that would be selected. But more importantly, you could put this together in a model that can account for all of this, where now we track kind of arbitrary number of strains of virus, arbitrary kind of mutations between them. And in addition, we want to take care of the fact that this is not going to cure the infection. We have some latent infection around. And so even when someone's on therapy for a while, virus is temporarily suppressed, there's always still some around, so that if at some point later someone is missing therapy, that could come back. So just to show you like an example simulation here, how we build these integrated models, and in each step here, there's a sort of a different source of data that's informing this. So we could choose some drugs we want to simulate, look at the drug levels in the body over time based on the pharmacokinetic parameters of the drug. Then we might consider a particular adherence pattern. There are surveys and other measures that try to estimate this at a population level, the whole distribution of adherence that people have to therapy. Could combine this with these pharmacodynamics, that's these dose response curves. That would tell us what the fitness of the virus is over time. And then using this mathematical modeling framework can kind of integrate that all to say, what do we expect viral levels to be over time? And how are they affected during these periods of missed doses? When we include, sorry, this is really mad at me right now. When we include resistant strains in there, we see that they have different fitness levels depending on the drug because they have a different dose response curve. And here's an example of a situation where at some point here, a resistant mutant was generated during a period when a dose was missed and the wild type could replicate. It took a long time for it to grow to a detectable level because actually drug was taken pretty well there. Then during this time period, the resistant strain was able to grow up, helped by some other missed doses, and eventually reach high levels such that even when good adherence was resumed, the population was now resistant, and this would constitute a treatment failure. So we use this to understand a lot of different HIV drugs. This is just some examples, kind of a panel of like 25 different HIV drugs to understand how these patterns would look. Here's just some examples. Roughly, we saw that we would expect a few different classes of behaviors here, where for some drug, no matter how much you take it, resistance is kind of expected to be inevitable. Other drugs where high adherence associated with good outcomes, green here means therapy success, lower adherence associated with failure and the generation of resistant strains. That kind of window, like how adherent you need to be to have a good outcome, varies by drug. And there are other drugs that even though resistant mutations exist, there's actually very unlikely to select for them, and that when someone fails therapy, it's more likely to be just because wild type virus grows back. And all of this is kind of related to the shape of the dose response curves for those drugs and how sharp their pharmacokinetics is. Actually, in these cases, if when you miss a dose, the drug just drops to super low levels right away, that's actually, in a sense, better because it doesn't leave much time to select for resistance. So that led us to actually trying to understand how this might impact a new really exciting new avenue for HIV therapy, which is long-acting therapies. So the idea here is that we want to make it easier for people to take HIV drugs. Let's not have a pill you have to take every day. Instead, very genius bioengineers are coming up with many different ways people might be able to take drugs. So anyway, from just making new like nanoparticle formulations of drugs, even just making better chemical structures that are cleared less quickly from the body, having kind of microarray patches that slowly deliver blood through these microneedles that you can't even feel. There are injectable drugs that like deposit in sort of subcutaneous tissue and slowly diffuse through your body. This is a vaginal ring that sits and slowly releases drug, and then some subdermal implants, kind of like a type of birth control that you might have heard about, to also slowly release drug. So one big worry behind these therapies is something that's honestly to this day like limiting their deployment in many parts of the world, is this worry about resistance. So we think that the idea of long-acting therapies is just to stretch out these pharmacokinetics. Like everything else is the same about the drug, you just make it farther away from the peak and the trough. So now you're just taking it once every few days. But the worry here is what happens if someone, stops taking the therapy at some point, be it for a short or long time. Are we now in a regime where we might be selecting for resistance? And is that regime kind of longer in time when you have a slow release drug versus maybe only lasting a few hours here? And maybe it doesn't even require missed doses. Maybe this could even happen during regular time between doses. Okay, well, I'll just do it this way. Oh, okay. Anyways, so we were interested in looking at this. This is something that Morgan and I worked at when we were both working together at Harvard. And we actually started this whole thing because we met some bioengineers who were trying to make something like this. This was a drug that was implanted in the stomach and slowly over a week released the therapy. And so we were interested in helping them understand whether this would be beneficial to alter the pharmacokinetics this way, or whether there was a risk of resistance. So we were able to put this together in a model and do simulations, taking into account a whole population level distribution of adherence levels to really try to understand, would the distribution of people that's succeeding or failing therapy be a lot different when you had this type of extended pharmacokinetics. And in particular, for one drug, it's called dolutegravir. It's kind of a newer generation HIV drug that's pretty good. So interestingly, in this case, we actually found that with the long-acting therapy, we really saw very minimal differences in the predictions of the risk of resistance for individuals taking this, even so much that we could reduce potentially like the peak dose that was given and still have very similar outcomes when you had this long-acting therapy. The way that people missed and picked up missed doses ended up mattering. Like if you have a weekly therapy and you miss it, do you not come back to the next week, or do you just get to take it the next day? That's the difference between these scenarios. And in this case, the result was pretty promising in that we don't actually expect things to be worse with this therapy. But we don't know how generalizable this is. This is one, like what we thought, very promising approach. But overall, people are developing many, and in particular, one that's pretty far along is actually a 2-month injection that individuals can take. It's developed, it's actually FDA approved, but still hasn't reached a very large audience. And so one of my then PhD students wanted to understand more generally, when you make therapies long-acting, when is it that they're going to make resistance risks worse, or is that rare, or is it common, and maybe that just was an odd case of that drug that we studied. So she was able to do a lot of nice work on this, which you can't see. Oh, okay. So yeah, so she looked at this sort of a whole range of different drug levels, from giving it every day, every few days, every week, every month. And she looked at the impact of where is the resistance coming from. So in this case, different places resistance can come from. Maybe it's pre-existing. Someone actually already has that resistance mutation at a low level before therapy, or maybe it doesn't exist. That just depends on the individual, their previous therapy history. And so she was able to look at, as a function of as you make drugs have longer and longer and longer half-lives, what's the risk of resistance? How does it change? In this case, she looked at if a strain is really, really resistant versus just partially resistant. In one case, sort of no impact of having a long-acting therapy. In another, it actually decreased the chance of resistance. When she looked at resistance mutations that didn't pre-exist but had to be acquired. This is really slow. Again, she saw that increasing the half-life actually made it less likely to develop resistance. And she looked at how this is the end, I promise. Then she looked at how this depended on what the drug kinetics were. We looked before at really fast absorbing drugs. Some of these methods are more slow absorbing in that it takes a while to reach the peak. In that case, she found sometimes increasing the dosing period actually made resistance risks higher. A lot of this depends on what can happen early on in the treatment when there's still a lot of virus around from the wild type. And she also looked at what happens if latent virus reactivates when someone is on therapy after many, many weeks and resistance actually arises that way. She also found in that case, long-acting therapies made resistance more likely. So overall, the results are quite complicated, basically depending on a lot of different properties of the drug and what resistance is available to it. Sometimes long-acting therapy makes resistance more likely, sometimes less likely. And really, it's not something you could make a general statement about and say we should or should not use these classes of therapies. We really have to do careful evaluation for each scenario to make a cost-benefit analysis. And in some cases, these are more robust to adherence problems, and in sometimes they're less robust to adherence. That also varies. So yeah, I will sort of just wrap up now. I just wanted to say a few things. In general, kind of breezed over how do we actually simulate all these models. I showed you some differential equations. We actually don't commonly just use differential equations for this. And the reason is that we have a lot of processes here, particularly mutation, which are very kind of rare and slow events that cannot be accurately simulated that way. Overall, in this model, we have some things that happen very quickly and with large population sizes, other things that happen very slowly. And so we actually develop some kind of custom hybrid stochastic deterministic models to simulate them with, which I'm happy to talk about later if anyone's interested in. But this is like a general problem in math bio. How do you effectively simulate reactions that you have processes occurring at totally different time scales? You want to be accurate, but you want to also be fast because we have to simulate like thousands of possible patients and outcomes and billions of viruses in each case and lots of different types of behaviors they might have in terms of drug taking. And we can also calculate a lot of things for these models to make things simpler in terms of like chances of establishment of mutations with some approximations that in many cases are pretty good. So yeah, I will end here. There are lots of different new therapies coming out for HIV. We also try to build models to understand many of these different therapies, and there are different challenges to doing that. People are trying to target the immune system more for HIV now. And if you've ever heard Morgan talk, I'm sure you know that modeling immunology is a whole nother ballgame that is really very tricky. So besides all the people I mentioned, these are members of my current lab who have helped out with various projects here over the years. And thanks especially to our funders of all of this, which have included the NIH, the Gates Foundation, and more recently CDC and NSF. Thank you.
So do we have questions that somebody wants to start with, or maybe I'll jump in? Okay, so I have a question, which is related to in cancer, there's this idea, or there have been reports that also exists in antimicrobial resistance about adaptive therapy, where you have a biomarker of success, and then you take treatment breaks, because the idea is that if you deplete all of the sensitive type, you're going to lead. what are your thoughts about this in HIV?
Yeah, so it's been a while since I really went through all the details of this, but there was a very cool conference that brought together people studying cancer evolution and virus and bacterial evolution to talk through all of these things. And it's very cool because there are a lot of things in common, but also the devil is in the details. There's a lot of things that are very specific to the system. This is just a good lesson for everyone. You know, if you're studying evolution in population genetics versus in cancer versus virus, like, it's good to know big principles exist, but that doesn't mean they like determine the outcome in your system. And so I think, yeah, in many cases, I don't think adaptive therapy would work for HIV. So obviously, like 0 with order, the difference between infectious diseases and cancer is you can spread infectious diseases. So having a time period with like intermediate viral loads is not, and not knowing about it is not good. So yeah, because you can transmit to other people. And also, you know, most HIV treatment cannot be done in the context of like a massive medical center where people are receiving cancer treatment, right? The idea is we need to like use this therapy in the field. So we can't be like monitoring everything people are doing. But I think like whether these therapies are beneficial or not depends on some intricacies in terms of how likely mutations are as a function of population size, obviously way more likely in HIV than cancer, and the strength of this competition. So I think there's really strong competition for resources among like solid tumors, for example, whereas in the context of virus, like, yes, they are competing for target cells, but that competition only kicks in when viral levels are very high. You know, for most of the time, there's sort of so many CD4 cells that it's just not relevant. So that's part of it. But I think there's probably more nuances to this that would be worth looking into as well. I think the results in cancer are very cool. I mean, I think we do need to think about ways of controlling evolution. It's definitely something we have also thought about. Like a lot of our work has sort of shown that things that are very bad for evolution include times when you have one drug at a high level and one drug at a low level, or if you have drug levels that are kind of heterogeneous throughout the body, and you have some drugs high in some body compartments and some drugs high in other body compartments, that's also very bad. So it's almost like you have to get rid of all of those things instead of creating them for most cases, I think, for viral evolution.
This is actually something I wanted to ask too about vaginal rings, because in so yeah, drug pharmacokinetics are going to vary in the body.
Yeah.
And if you have a tissue level of absorption, you may have different pharmacokinetics.
Yeah.
So are vaginal rings, I guess, used primarily as a transmission blocker, and then there's a switch?
Yeah, but I would actually love to look at this. Those actually like progressed through this whole pipeline during COVID when we were like not so much working on this and working on other things. So I haven't seen any modeling of them. It doesn't mean it doesn't exist. I would love to use that as an example of combining the PK with the models, because yes, they are primarily used to prevent infection. So the idea is that like they would be at the site of infection, virus would block there, wouldn't get anywhere, but no prevention is perfect. And, you know, people can be imperfectly adherent even to sort of implants or exoplants, whatever you call that class of drug. So yeah, if you have breakthrough infection, like someone didn't know they got infected and are still on this, and then that's like, in general for HIV prophylaxis, that's the big worry. So I think, yeah, it would be interesting to see like what you would expect in those cases, and just, you know, what are ways to just be more careful about that. Again, I want to clarify that, you know, resistance is not a reason not to treat. Someone having drug resistant infection is no worse than them having non-drug resistant infection. So it's not that our goal is to, you know, overall suppress the viral loads of the world, not to just prevent resistance. But if we can do both, like that would be the ultimate. So yeah, I think that's a very interesting example to look at in this framework.
I think that was good.
Just making sure. Some people.
Are there other questions? Well, yeah.
I think the two big ones. The first one was how much does individual level variability impact with the predictions you're making? And the second one's, I think there's a nice paper in the BMB, which was on one of their podcasts, that looks at drug adherence, and if you can get to take the particular drug one day, whether you should take it twice the next.
Right.
Is that relevant here?
Yeah, it's pretty interesting. Yeah, so I didn't go into it all here. And in earlier like versions of these models, we were actually kind of using population averages for everything in terms of pharmacokinetics and pharmacodynamics, but what we're kind of doing now is trying to incorporate like population variability at all of those stages to see the driver is, because I think that is probably very important. Like some percent of people who are developing resistance, they probably have different I think pharmacokinetics is like mainly the thing more so than pharmacodynamics, that they just they're not having drug absorption to the same levels, and understanding how much that is contributing, I think is really just and just to like properly like integrate over all levels of uncertainty here in these predictions. Important.
We've got poor absorption for increased clearance.
Yeah, true. Yeah, no, agree. That's just equally likely, I think.
Great. And then, yeah, sure.
Well, I have a question. So during the viral evolution, have you considered other mutations?
What sort?
Some virus have higher infectivity, something like that.
Oh yeah, I mean, that would still be viral evolution,
Yeah.
just for a different trait. Yeah, certainly we know HIV evolves in all of these ways. Like there are mutations that impact both its infectivity within individuals and in a population. People have looked at how the virus has evolved over time by looking at like how the population distribution of viral levels has changed over time, like of untreated people. And similarly, we know the virus is evolving to avoid immune responses. So it's not that you don't have an immune response to HIV, it's just you don't have a good enough one. And one of the reasons why it's not good enough is because the virus evolves very quickly to escape from specific anti-HIV immunity by antibodies and killer T cells. So yeah, that is happening for sure.
So can the virus have both mutations? One is to increase infectivity and the other
Oh,
one.
I mean, yeah, there are like the HIV genome is like 10,000 base pairs long. And so, you know, you can have like 4 to the power of 10,000 different viruses. No, not that much, but like there's insane amounts of variability, yeah, in the virus. Like every virus, you have a billion different viral particles in your blood at once. Like if you have untreated HIV, every single one of those is probably genetically distinct.
No, if not, well, thank you very much.
Thanks,
Alison.
everyone.
And beside us, in the lounge, and we're